So good afternoon. My name is Karambod Siddiq. I'm the director of the UWA Institute of Agriculture. Those who doesn't know me, welcome to the special seminar. But before that, I would like to acknowledge our country. The University of Western Australia is situated on the Noongar land. The Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. So today, we have a special speaker. So this Institute of Agriculture doesn't hold regular seminars. We have mostly public lecture whenever we have what we call occasional special seminars. So today, I would like to welcome Dr. Sruthi Narayanan. She's an associate professor of crop ecophysiology at the Clemson University, South Carolina, which is a small country town in the US, specializing in resilience mechanisms of agronomic crops under abiotic stresses. So how did I know Sruthi? Sruthi is an ex-supervisor, or always a supervisor, PhD supervisor, Professor Vara Prasad, which you probably all know, distinguished professor at Kansas State University. He was Sruthi's supervisor for master's and PhD, done some wonderful work. So that's how we met each other. But there's also another one which is not mentioned in my notes. Sruthi did her undergraduate in ag science from the same university where I did my undergraduate studies, although different times and era. Sruthi research program focuses on developing innovative strategies to establish climate resilient production systems that achieve the delicate balance between economic viability, that is the profitability, and we have a farmer here, Rod Birch, I think only one farmer. In the face of climate change, this work is critical to supporting sustainable agricultural practices, moving toward climate-smart agriculture and climate-resilient crops. Her research and teaching programs have been recognized nationally. Sruthi received several awards, including the Crop Science Society of America Early Career Award, the highest honor for a young scientist in the US in agriculture science. She has also served in multiple leadership roles within professional societies and academic committees, and is currently the chair-elect of the Crop Physiology and Metabolism Division of the Crop Science Society of America. So they have different divisions, which is really good for early career researchers to work along with the senior people and get that leadership, mentorship, and so on. Today, Sruthi will explore sustainable agriculture in the omics era, multiscale physiology to breed climate-resilient crops. Please welcome Associate Professor Sruthi Narayanan.
Thanks.
All yours. You got all that. Is that here? Okay. Thank you.
Yeah, thank you. Okay. Thank you, Professor Siddiq, for the nice introduction. And also thank you for inviting me for offering this lecture. I'm honored to be here today. And good afternoon, everyone. As Professor Siddiq mentioned earlier, I'm a crop ecophysiologist at Clemson University in South Carolina in the United States. And today I will talk a little bit about our research program that focuses on multiscale physiology to breed climate resilient crops. So here's an outline of my talk for today. I will start with a bit of background. I will talk about the big picture and the grand challenges that we focus on, and where the Clemson Crop Ecophysiology Lab fits in. And then I'll talk a bit more about our research program, its vision and goals. And then I will elaborate 3 specific lines of research in our lab: our lipid research, root research, and sustainable agronomy research. And then I will wrap up my talk with a few concluding remarks. So let's first start with the food gap. What you're seeing here is a depiction of the food gap existing in the world. So we expect that by 2050, world population might reach around 10 billion. And to feed that large population, we will need to increase food production by 55 to 60% or so. That's a huge number. and that's a huge challenge too. Climate change magnifies that food challenge. What you can see here is the percentage change in crop yields by 2050 due to climate change. The red color indicates decline in crop yields, and the green color indicates increases in crop yields. And we can see that most, if not all, crop growing regions around the world are highlighted by some shades of red only, indicating significant crop yield losses due to climate change. As many of you would be well aware, two major stresses by which climate change is impacting our crops are heat stress and drought stress. On this map, you can see the water stress predicted for different countries in the world by 2040. Again, a red color indicates high level of water stress. And you can see that the entire United States and Australia and most crop growing regions in Asia and many in Africa are highlighted by dark red only, indicating very high level of water stress. Global warming is another challenge. It will increase the intensity of heat stress on our crops. And on this graph, we can see, or we can get an idea about the yield losses that we need to expect for our major agronomic crops. Look at the numbers. They are huge. So in our lab, we work toward, or we want to work for developing climate resilient crop varieties that will enhance the sustainability and profitability of our production systems. So how do we put our goals into action? Here are our approaches. So we study physiology at multiple scales, the cellular level, whole plant level, and whole field level. And our goal is to identify physiological traits and mechanisms that will contribute to climate resilience or abiotic stress tolerance in crops. And then we try to capture the genetic diversity for the traits of interest. And here we screen germplasm collections, including mapping populations, breeding lines, cultivated varieties, wild relatives, etc. And we employ state-of-the-art physiological techniques and high-throughput phenotyping approaches to capture the genetic diversity for the traits of interest. And then we move on to integrating phenome to genome. And here we try to identify the genes and genomic regions regulating the traits of interest so that we can use them as molecular markers for the traits of interest and to incorporate those traits in breeding programs. Finally, we scale up to the cropping system level. And here we focus on identifying sustainable management practices to enhance the performance of our crop varieties in the production systems and also to enhance the sustainability of the cropping systems as a whole. So for doing that, we apply principles from various disciplines, from agronomy through various omics techniques to plant breeding. So ours is an interdisciplinary program. So we work at the intersection of agronomy and plant breeding with three major focuses: enhancing climate resilience, improving resource use efficiencies, and minimizing environmental risk. So these three are the three pillars of our research program. And the information generated from my research program is informing my teaching program too. I have 50% teaching appointment, and I teach multiple courses related to crop science. So in the coming slides, I will talk about specific research projects in each of these three research areas. And I'll start with the first one, enhancing climate resilience. And here I will talk about our lipid research. And in this research, we are exploring lipids as breeding tools for improving crop heat tolerance. And these are my graduate students who conducted the research projects that I'll be talking about in the coming slides. So, why lipids? As some of you would be aware, cell membranes are prime targets of heat stress. So heat stability of membranes is fundamental to heat tolerance at the cellular level. The stability of membranes in turn depends upon the structure and composition of the component lipids. So we hypothesized that heat-tolerant genotypes will remodel their lipidome under heat stress to ensure functional membrane homeostasis. And we tested the hypothesis on multiple species, including soybean, peanut, and wheat. And in the coming slides, I will talk about the results from our peanut study. So in this research, we used a subset of a recombinant inbred line population, and we grew the plants under controlled environmental conditions. And we maintained the plants at optimal temperatures until flowering. And after flowering, or at the onset of flowering, we started the heat treatment. And the duration of heat stress was for 14 days. First, let's look at the impact of heat stress on leaf physiology. Here I have included two traits which are closely related to leaf photosynthetic rates. And they are chlorophyll index and efficiency of photosystem 2. And you can see that both traits significantly decreased under heat stress conditions, indicating significant negative impact of heat stress on leaf physiology. Now, let's look at the changes in the leaf lipidome. Here I have presented the changes in unsaturation indices for the major lipid classes we detected in the tested peanut population. If you're unfamiliar with unsaturation index, it relates to the presence of double bonds in the fatty acid chains of lipid molecules. And you can see that unsaturation index significantly decreased for most lipid classes. And that was the primary adaptation mechanism to heat stress at the lipidome level. And that is to maintain optimal fluidity and integrity of membranes under heat stress conditions. If you're unfamiliar with that mechanism, I have a slide here to explain that. So, as I mentioned earlier, unsaturation refers to presence of double bonds in the fatty acid chains of lipid molecules. And in plants, the fatty acid double bonds are primarily in the cis configuration. The cis double bonds introduce bends or kinks in the fatty acid chains of lipid molecules, which reduces the degree of ordered packing or close packing of lipid molecules within the membranes. In other words, higher level of unsaturation leads to increased disorder within the membranes, and that will lead to increased fluidity of the membranes. High temperature also leads to increased fluidity of the membranes. So one way by which plants can adapt to high temperature stress is by decreasing the degree of unsaturation or the presence of double bonds in the fatty acid chains, and thereby maintaining the optimal fluidity of membranes under heat stress conditions. We also wanted to test how various lipid classes achieved a lower level of unsaturation under heat stress condition. So what is the mechanism? So we tested the changes in the individual lipid species level. And here I have presented the species level changes for a major leaf lipid class. It's a plastidic lipid too, and that is DGDG. What we can see here is that the DGDG species containing the highly unsaturated 18,3 acyl chains significantly decreased under heat stress conditions, and the DGDG species that contained saturated or less unsaturated acyl chains significantly increased under heat stress conditions. And then you can imagine how those type of changes will lead to an overall decrease in unsaturation index of the DGDG lipid class. Similar changes were observed for the other plastidic lipid classes and extraplastidic lipid classes as well. So the primary lipid alteration in peanuts under heat stress was the removal of the polyunsaturated fatty acid chains from the membrane lipids. But what is happening to the polyunsaturated fatty acid chains removed from the membranes? Where do they go? So we found that there was another group of lipid molecules which were also highly heat responsive, and they were triacylglycerols. As you can see here, tags or triacylglycerols clearly separated the temperature treatments. When we looked at the individual species level changes, we found that tags containing the highly unsaturated 18,3-acyl chains significantly increased under heat stress conditions. And that supports the recycling power or ability of tags. So tags can act as recycle lipids in plants or buffer lipids. They can take up the fatty acid chains expelled from the membrane lipids, and by doing that, they can adjust the unsaturation indices in the membrane lipids. So our results suggest a possible role for tags in heat stress adaptation in peanuts. That is by taking up the acyl chains expelled from the membrane lipids and thereby maintaining optimal fluidity of membranes under heat stress and making the plants more heat tolerant. We found that there was another group of lipids which also took part in the acyl sequestration mechanism, and they were phytosterols. And to give you some examples, here I have shown 2 cytosterol species containing polyunsaturated acyl chains. And look at the huge increase in the levels of these polyunsaturated cytosterols under heat stress conditions. So our research suggested the possible role for phytosterols as well in the heat stress adaptation of peanuts. This lipid biochemical pathway map places the observed heat adaptive lipid remodeling in the broader context of plant lipid metabolism. The major takeaway from this diagram is the movement of the polyunsaturated acyl chains from the plastidic lipids and extraplastidic lipids to the neutral lipid buffers such as TGs and SEs. And that was the primary heat adaptation mechanism in the tested peanut population for maintaining optimal membrane fluidity and integrity under heat stress conditions. So far the trends that I presented were at the population level. So we pooled data across all the genotypes in the tested population, and I presented the population level trends. Based on the observed heat adaptive lipid remodeling, we were also able to identify the most heat tolerant and most heat susceptible genotypes. On this hierarchical clustering map, you can see how different genotypes were clustered based on heat tolerance or heat responses. And this was the most heat tolerant genotypes cluster, and this was the most heat susceptible genotypes cluster. And all these genotypes, the tolerant ones, had the lowest levels of the 18,3 containing plastidic and extraplastidic lipids, and the highest levels of the 18,3 containing tags and SEs. To verify our results, the heat tolerant parent ICGS 76 was also grouped in the same cluster. On the other hand, this was the heat susceptible genotypes cluster, and these genotypes or RILs had the highest levels of the 18,3 acyl containing plastidic and extraplastidic lipids, and the lowest levels of the 18,3 acyl containing tags and SEs. Again, to verify our results, the heat susceptible parent for the RIL population was also grouped in the same cluster. We also looked at the changes in the expression of different genes related to the observed heat adaptive lipid remodeling. So these 2 genes, PDAT and DGAT, some of you would be familiar with those enzymes or genes, they are responsible for the synthesis of tags. And what we found was that the expression of both genes significantly increased under heat stress condition in the tolerant genotype compared to the susceptible genotypes. So that confirmed the role of tags in heat stress adaptation in peanut. And these 2 genes are involved in the synthesis of sterol esters. And I have included some more information here regarding how they are involved. And what I want to emphasize here is that the expression of both these genes significantly increased under heat stress condition in the tolerant genotype compared to the susceptible genotype. Again, that confirmed or verified the role of SEs in heat stress adaptation of peanut. We were also able to identify a genomic region associated with the heat adaptive lipid remodeling. And that region mapped to chromosome 16 of peanut, and it included 15 SNPs. And we will use that information for identifying potential QTLs for heat-tolerant lipid remodeling in peanut. So, as we saw earlier, the unsaturation index of membrane lipids decreased under heat stress, which was the major adaptation mechanism at the lipidome level. And we discussed the acyl recycling mechanism or acyl sequestration mechanism as a major reason behind this trend. We also wanted to test whether the activity of enzymes responsible for unsaturation was also altered. So in plants, the enzymes that introduce double bonds in the fatty acid chains are known as fatty acid desaturases. Several fats or fatty acid desaturases have been reported in peanuts, and here I have shown some examples. As you can imagine, fats have substrate specificity. So specific fats act on specific fatty acid chains only. And we wanted to test whether the activity of specific fats are different in peanut genotypes contrasting for heat stress responses. So we tested that in a field study, and we used these genotypes in that particular study. So those genotypes were selected based on their contrasting responses to heat stress or drought stress. And we imposed heat stress during flowering using heat tents. If you like to know how our heat tents work, here I have a video showing that. So the heat tents are movable over rails. And whenever it's time to impose heat stress over on specific plots, we can move the heat tents, and when the heat tents reach the designated place, we halt it there, and then you can close the side walls and end walls, and heat will build up in the heat tents. So our heat tents work based on the radiative heating. That means it heats up due to solar radiation. Okay, and what we found was that the expression of the FAT32 gene was significantly lower under heat stress condition in the heat-tolerant genotype SPT0607. But that trend was not noticed for any other genotypes which were either heat-sensitive or less heat-tolerant than SPT0607. That indicates the role of FAT3s in heat stress adaptation, because FAT3 introduces a double bond in the 18-2 fatty acid chain, and it makes it an 18-3 acyl chain. So reduced FAT3 expression means reduced conversion of 18-2 to 18-3. So lower level of 18-3 acyl chains, lower level of unsaturation index, and better heat tolerance. So to conclude, under heat stress, the adaptive mechanism of the peanut leaf lipidome were the removal of the 18-3 or the polyunsaturated acyl chains from the membrane lipids, which were sequestered into tags and SEs. And that change led to decreases in the unsaturation index in membrane lipids. We found that reduced FAT3 gene expression was also taking part in the reduction in unsaturation indices. And we were also able to identify a region on chromosome 16 as associated with the heat adaptive lipid remodeling. And we are currently exploring this information further to identify or develop potential QTL for heat tolerance. With that, I'm shifting gears, and I'm moving on to the second research area that I want to talk about in today's lecture. And here I will focus on our second research goal, which is maximizing resource use efficiencies. It's a totally different topic. I'm going to talk about our root studies a little bit. And these are my graduate students and postdocs who did the work that I will be presenting on the coming slides. I know that many of you are working on roots, so you would be familiar with that. But for those of you who are a little bit unfamiliar with this topic, I will give you a little bit of background information. So what makes root breeding so critical? Well, there are so many reasons, as many of you would be aware, and I will talk about one. So you would be aware about this metric, water use efficiency, which is an important trait in crop improvement programs. It's a highly sought after trait by the crop industries. For example, the US cotton industry has set a goal for improving water use efficiency in cotton production by 18%, which is a very ambitious goal. Similarly, water use efficiency has always been a priority by the Australian cotton industry. So it is a highly sought after trait by the crop industries. And these are three targeted mechanisms for high water use efficiency in crop breeding: optimizing water absorption by roots, enhancing carbon assimilation, that is enhancing photosynthesis and transpiration efficiency, and partitioning more assimilates into the harvestable part, basically increasing harvest index. In this particular project, we are focusing on the first one, optimizing water absorption by roots. We are specifically targeting to identify specific root architectural and morphological phenotypes or traits that will enhance root access to water and conservation or saving of soil water for late season use. And we are specifically focusing on identifying molecular markers for beneficial root traits. So why molecular markers? As some of you would be well aware, incorporating root traits in breeding programs is very challenging. A main reason is that our current root phenotyping methods are highly time consuming and low throughput in nature, very energy intensive, money intensive, labor intensive, so generally very low throughput in nature. So in order to incorporate root traits in breeding programs, we need high throughput measurement techniques for root traits. And molecular markers can be explored as high throughput selection criteria for associated root traits. And we conducted our root research on multiple species, including soybean and cotton. And in the coming slides, I will present results from our cotton study. And in this research, we used a subset of the US upland cotton diversity panel. The entire diversity panel is a large collection. It has 382 accessions. But in this particular research, we used a subset of that. The subset, which is known as a core set, includes 22 accessions, but it represents 82% of the total allelic diversity of the diversity panel. So currently it stands as a golden collection for dissecting the genetic architecture of quantitative traits in cotton. So that was the population that we used in our studies. And we conducted the study under field conditions. We found significant genetic variability in above ground traits in the core collection in terms of biomass, seed cotton yield, lint yield, water use efficiency, leaf area index, crop water use, etc. Now, water use efficiency, as some of you would be aware, is the ratio between biomass and water use. So it can be increased by increasing biomass, reducing water use, or a combination of both. The strategy of increasing water use efficiency by reducing water use often results in plants with poor yield or reduced vigor. So in that case, the metric water use efficiency has no practical value or meaning. But in our case, what we found was that water use efficiency was either not related to crop water use or soil water depletion, or it only had a weak negative relationship with soil water depletion. On the other hand, water use efficiency was having a strong positive relationship with shoot biomass production. So that indicated that in the tested population, increased shoot biomass rather than reduced water use increased water use efficiency. And that enhances the value of the water use efficiency metric for cotton breeding. And this scenario can result primarily under two cases in agronomic crops. One, some genotypes produce higher biomass than the other genotypes, but with the same amount of water use. Two, the larger values for increase in biomass offsets the increases in water use of genotypes. Now, let's look at how root traits contributed to these traits. And here is how, this is how we measured root traits in our study. So we used an in situ root imager to measure roots. It was the CID root imager. So for using the imager, you need to install the access tubes in the crop rows, and the access tubes stay there. They are clear acrylic tubes. And whenever you are ready to measure root traits, you go to the field with the imager and you insert the imager into the access tube. The imager takes 360-degree turns and it captures images which you can analyze using particular software that comes with the CID instrument. So this technique allows us to measure root development in situ, non-destructively, at regular intervals throughout the crop growing season. And what we found was that the genotypes differed in terms of root length distribution in various soil depths. So here we can see the distribution of root length in different soil profiles in different crop growth stages. The different colors on the bars indicate different soil depths. For example, blue color indicates the shallowest depth and the yellow color indicates the deepest depth. And what we found was that the genotypes with the best above ground performance also had the highest distribution of root length in the deepest soil profile. And on the other hand, the poor genotypes with poor above ground performance also had the worst amount of root length distribution in the deepest soil depth. We also looked at the correlation of root length at various soil depths with shoot biomass and water use efficiency. And what we found was that the water use efficiency and shoot biomass was positively related to root length in the deepest depth, but they were not having any relation with root length in the shallower depths or with the total root length. So these results indicated the importance of deep rooting for improving water use efficiency of cotton in our production conditions. And our results suggested two contrasting root architecture models. The first one, which you can see here, refers to increased root distribution in deeper soil profile, as you can see here. And that helps for better resource capture from the deeper soil profile, which will enhance above ground growth, dry matter production, and water use efficiency. The contrasting model you can see here, and that is characterized by reduced root length distribution and reduced total root length, reduced total root length and root length distribution in the deepest profile compared to the other one. And this type of a model will be associated with poor resource capture from the deeper profile and poor above ground growth and water use efficiency. And these results are supported by the steep, cheap, and deep root idiotype proposed for agronomic crops in literature. Many of you would be familiar with that root model, which is a very well-known model for root architecture in agronomic crops. So here, steep refers to steep root growth angle, which promotes deeper rooting. And cheap refers to reduced metabolic cost of soil exploration. So here, what it indicates is that it's not about producing a whole lot of roots, not about working harder, but working smarter. Invest in only that part of the root system which will be of maximum advantage to the plant. So that is what is going on here. And we were also able to identify some SNP markers linked to deep root length in cotton. And we are further exploring this information to identify molecular markers for beneficial root rates in cotton. And currently, we are exploring the larger diversity panel for traits that will increase water use efficiency. And we will also do a GWAS analysis to identify molecular markers for beneficial root traits. So with that, I'm again shifting gears, and I'm moving on to the last research area, minimizing environmental risk. And here I will talk about our cover crop research. And these are my graduate students and postdocs who conducted the research projects that I'll be talking about on these slides. So, industrialized agriculture, as we all know, has revolutionized the way we do farming, or even the way we think about farming. And it has boosted crop yields quite a bit. It shifted our focus from yield to even more yield. But we have realized the impacts. And what you can see here are dust storms in various parts of the United States all through these years. We also realized that diversity is the answer for many of our questions. We now know how important crop diversity is to enhance ecosystem services such as soil health or pest control or cropping system resilience to climate change, like that. And an approach to enhance diversity is cover cropping. And cover crops, as many of you would be aware, can bring a myriad of benefits to our farms. And some of those examples are given here. But in order to get the best benefit out of cover crops, we need to optimize the system very well, because cover cropping is highly region specific. What works in my farm may not work in your farm, like even though we are adjacent to each other. So we started our cover crop research back in 2015, and then we expanded our program through these years with larger federal funding. And currently it's part of Clemson's historic $75 million USDA grant for sustainable agriculture. This is a huge project. We are 29 co-PIs working on that project. And this is a large project with collaborations among multiple universities in South Carolina, which is the state from where I'm coming. And we have collaboration with extension institutes, nonprofits, and industries like that. And all research part of this project are done on farmers' fields only. We have 450 farmers enrolled in this project. And in our lab, we are specifically investigating our objectives on 20 plus peanut farms. And other co-PIs are working on other farmers' farms too. So in our research, we are looking at optimizing cover crop benefits through management and choosing the right system. In terms of management, we are looking at different things like cover crop selection, planting time, termination time, seeding rate, seeding method, etc. And in terms of the system, we are studying different systems like soil cover cropping or interseeding or living mulch systems. In the coming slides, I will talk about our interseeding research. So if you're unfamiliar with interseeding, here I have a picture showing that. So these are our cover crop interseeded fields, and our cash crop here is corn. And as you can see here, cover crops are sown in the inter-row spaces of adjacent corn rows. So if you want to see, oh, okay. Okay, I had a video, Anna, here. I think yeah, after this one. It's okay. Yeah, I'll just move on. I had a video showing how interseeding is done and how our interseeder works, in case you wonder how this is done, because that's a question most people ask me. How do you do this? Because when corn is there, how do you plant your cover crops? But we have machine for doing that. The key part is that the timing, when you interseed. It shouldn't be when the crop is too tall or too small. So we did some research in terms of the optimal time of interseeding. But anyway, that was the video about, but I can continue, no problem. So now, why interseeding? What was the motivation to look at interseeded system? And that is going back to the particular climatic condition or environmental condition back in the southern US, where our research is happening. So in the southern US, we have long crop growing season from April to November. So to give you some perspective, our summer or spring starts in somewhere in March, and then comes summer, and then by September or so, temperature cools down and it's fall season, and November, December, winter, January, February, peak winter. So, but the day length is longer and the temperature changes are actually mild, so farmers can take long crop seasons, and it usually ranges from April to November. So if a farmer is harvesting their crop by November, and if they want to plant cover crops, they will be planting cover crops very late. And then comes winter. The cover crops may go to winter dormancy, but they restart their growth when the following spring comes. But then very soon, it's again time for planting your cash crop. So cover crops may not get enough time to establish and produce sufficient biomass for getting the best benefit out of cover cropping. So what would be an alternative? We thought about interseeding at that point. As you can imagine, it's a very risky system too. You're growing another crop along with your cash crop. So unless you manage the system very well, cover crops can act as weeds and reduce the cash crop yields. So the optimization of the system is very key for getting the best benefit. What we ideally look for is enhancing the complementarity between the components in the system and minimizing the competition between the different components of the system. So there are several things that need to be optimized before you can reap the best benefit out of this system. And in this particular research, we focused on these components: tillage, cover crop selection, seeding timing, and seeding rate. And I will quickly go through the results. So we found suitable cover crop species and their seeding rates that would enhance the performance of the corn production system that we investigated. Investigated in the sense, the system that we investigated was a corn cover crop interseeded system. So the results are specific to corn production systems. And we observed benefits in terms of soil water saving, weed suppression, corn biomass production, and corn yield. And the results were similar under reduced till conditions and conventional tillage conditions too. And now we are moving on to the next level. And in the current research, we are trying to focus on quantifying the benefits of cover crops for climate smart commodity production. So we are trying to explore cover crops as an approach for reducing greenhouse gas emission and enhancing carbon sequestration. To conclude, so far in our research, we identified some traits, mechanisms, and genotypes that we can incorporate into breeding programs. We were also able to identify some management practices that will enhance the performance of these crop varieties and the sustainability of the cropping systems. And all these efforts were directed toward three major goals: sustainability, profitability, and climate adaptability. With that, I would like to thank these grad students and postdocs and undergraduate students. They are not here in our lab. They did all the work, so everything done by them only. And I also want to thank our funding agencies, US Department of Agriculture, which is our major funding agency, and nonprofits and commodity boards and private industries, and our collaborators. With that, thank you all for listening, and I would be happy to take any questions if I have time.
Thank you, Sruthi. You have timed well, and so we'll take some questions. Be brief and tell who you are, so at least Sruthi know who is. So first question, Sneha.
Yeah.
Sure.
I'm Sneha. I just finished my PhD here and started working on nitrogen use efficiency.
Mm-hmm.
So when I saw about what it is, because my PhD was on drought stress, just was curious to know how did you like measure the water stress and the field conditions in cotton. Also the beautiful picture when you showed, I didn't notice, like, was there something we used to cover the soil, some mulching or something?
Yeah, so in the cotton study, we did not cover soil. It's like the same practice that our cotton farmers do. They don't cover the soil. So we did not cover the soil. Your other question, how did we measure water use efficiency? We used neutron probes to measure soil volumetric water content up to 1 meter. And then we had measurements of soil water content or volumetric soil water content at regular intervals. And then you can use the soil water balance equation to calculate crop water use. Yeah.
Come and attend my lecture for the fourth year students. Huyen.
I just want to agree with Sifan because I did the similar thing.
Okay, good. Yep.
Yeah, I'm on the Department of Primary Industries.
Mm-hmm.
The question I have is on the heat stress that you've done.
Mm-hmm.
So I realized some of the data you've collected from leaf samples of only 50 parts. And as we know, some of the reproductive and nutrition tolerance would be different. So I just want trying to see whether there is some work on the reproductive parts, so that's the main thing that we focus on.
Yes, good question. Yes, we did lipidomic work on reproductive tissues as well, primarily anther tissue. I have a couple of papers on that. I did not present that data here, but yes, we have done that. And the responses were similar. But in case of reproductive tissues, the more heat responsive lipid classes were phospholipids instead of galactolipids, as in leaves. Yeah, but the other mechanism were primarily kind of similar. There were differences too, but yeah.
Caitlin?
Yes.
Really nice talk. And
Thank you.
I think you did a really good job of outlining how your lab group have helped contribute to the growing body of knowledge. So, a really great job. I was quite interested in the temperature shelter that moved along the run.
Mm-hmm.
Curious to know if you can comment on how the rate of temperature change within that enclosure and how you control for other things like reduced sunlight covers and reduced wind speed and the likes, and how that might have an effect on the results that you found.
Yeah, so good question. The temperature difference, first part. The temperature increase was 10 degrees Celsius. And well, it's a range. So it varied from, we have the data presented in our paper, so you can actually see the data. We have graphs there. But anyway, it ranged from 8 to 10 degrees Celsius. So what we do is that when the temperature differences, when the temperature inside the heat tent is more than the threshold value, we roll up the side walls. So the temperature goes down. And then when the temperature reaches the minimum threshold, we roll the side walls down. So temperature again goes up. So the temperature inside the heat tents will stay within the range that we set at the beginning. So that's how we set adjust the temperature inside the heat tents. Now, your other questions regarding sunlight. Yeah. So yes, that's a very important question. You actually need to use specific plastic for building the heat tents. All plastics will not work because they will be blocking some sunlight. So we did some trial and error, and we worked with private companies to identify the best material. So this one actually transmitted 95% of the outside solar radiation. So that's sunlight. And then your last question, I guess, was regarding wind speed. It will be like that only because winds will not be coming inside if the tent is intact.
Do you have little fans at all operating within the canopy enclosure? Or?
We did not have that in our tents, but some people use that also to regulate temperature.
And homogenize as well. So for the plastic, we have done other studies in Shelby Park, and we have got clear plastics like glasses. No problems.
Yeah. And our heat stress duration was only 14 days. So if it is like longer duration, then we will definitely need to have that arrangement. Yeah.
A lot of our, just a side comment, a lot of our problem is only 2 or 3 days is enough. Up to 2 hours is enough. So a lot of our studies in heat stress, what we do is that we take it up to 2 or 3 hours during the diurnal pattern, and then we relieve the stress. So that's which does a lot of damage to the farmers like Rod. The crop is looking good, the heat comes in 2 or 3 hours, then may continue next day 2 or 3 hours, then the damage is done. So, Doug.
Hi, Doug Hall, industry consultant. Putting climate change aside, I mean, your work can lead to new varieties that can grow into environments where it can't grow, so it gives a greater spread of genotypes for a farmer. Are you translating this into breeding, are you looking for varieties that have a wide plasticity, or are you looking for a series of varieties with narrower bands of plasticity which are applicable to different environments?
Yeah. So that's a good question. Currently, the varieties that my breeder collaborator produced were varieties where we incorporated specific gene or genes. So they were having 1 or 2 traits incorporated into the elite background. So these are like elite varieties plus some new traits added. So basically, they end up having a broad spectrum of traits that the producers are desiring for in our region.
And just for, I mean, you mentioned some fractionation of membranes, is that plastic? So have you got a sense of how plastic the different membranes are in response to these genes? And presumably plastics, polyphyletes are critical for production. There might actually have to be other membranes, and reference to the question about reproductive organs and their membranes. So have you got a sense of the relative importance of different membrane types in cells?
Yeah. That's a good question. I have not looked at it, or I have not measured it in our studies. But as I mentioned earlier, the type of lipids which were playing the major roles in leaves and anthers were slightly different. In the anther tissue, it was extraplastidic lipids, and in the leaf tissue, it was plastidic lipids. So, yeah, I assume that there might be some sort of changes. Who is playing the major role? Yeah.
England.
Hi,
Yes.
Nice talk. My name is Long Chen. We have meeting tomorrow.
Okay.
Just question about the youth research. You used the restroom that we call CI 603 or 602.
602.
Yes, that we have to bury the tube inside the
Yes, yeah, we
before
install the
we-
access tube.
So that's may cause question about a lot of questions went wrong that. And we know this is very common use. How about how do you confident about the roots? You know, sometimes only the roots touch the surface of the tube, so you can bend the roots. So what the position, depending on the position, you place your tube in the soil, and you can see it's collect data, root data in the soil.
Yeah.
So how confident about the lower part of the soil, the roots in that deep soil, can collect, represent the whole root system?
Yeah, that's a good question. The CI 602 instrument has its own limitations. And as you correctly mentioned, we will be viewing only the roots that will grow around the access tube. So that's a limitation. So I would say that the precise quantification of root system per genotype may not be accurate, but we have not done that. We were comparing the genotypes. So the relative comparisons will still work, but the absolute quantification of roots may not be accurate with this method. So for that, you need to
You need to use the
distrust
muscle
it.
power. About 30 years ago, I did a wonderful field experiment, the angle and everything. So finally, I gave up. The reasons which you mentioned, A is not going deep, it's an angle. So whatever roots come around that, that's only captured by the camera. That's like a colonoscopy. And then when you go deeper, it's not. And again, in her data, she only showed up to 50 centimeters depth. We know, depending upon the situation, the roots have to go deeper. But this is a method which we gave up, and then I used augering. And what you could have done is that you could have went and done some augering work and then do a calibration or a correlation. But
Yeah.
anyway, that's not the.
We actually have that data, which is presented in our paper.
Yeah, okay.
Yeah, so
But
we
this is a real
complement,
problem. It's a real
yeah,
problem.
the
And so I
data.
gave up to that. Nanti, and then we got Agaya. So now.
Nanti Waller from UWA. Fantastic presentation covering
Thank
all
you.
three aspects. I'm not a gene jockey or only person. The third component, cover crop, I could see that you are looking at some carbon dynamics. Are you looking at nitrogen dynamics, greenhouse gas emission, nitrous oxide?
Yeah, nitrous oxide and methane.
Okay, so you're looking at all the three, CO2. Well done, thank you.
Agaya.
Thank you for the wonderful covering a wide variety of topics. My question is around multi-omics, and with the rise of advanced technologies, we have a diversity of options to look for biomarkers for stress tolerance or even other complex traits. How many genetic markers are too many genetic markers for building and how do we select only the best options? Like, should we go for metabolomics? In metabolomics, we have options for multiple chromatographies. Should we go for proteomics? We have options for like different peptide selections. So yeah, what is your take on that?
Yeah, I would say that it depends upon what you're looking at. If it is like lipid biomarkers, obviously, we would be doing lipid profiling. If you are specifically targeting a protein or enzyme, and if you want to do protein quantification, obviously it would be proteomics. Or if you are looking at specific genes and how the gene expression varies, you can resort to other techniques like RT-PCR or things like that.
Sorry to extend.
It's okay. Yeah.
So my question was regarding QPLs associated with those markers. For example, lipid QPLs, for example, metabolic QPLs, and protein QPLs. So if we go into all of these omics, there will be a variety of QPLs coming up for specific traits. And how do you deal with that?
Oh, as a breeder? Yeah. Of course, incorporating too many traits in a breeding pipeline would be leading to so many challenges if your end goal is producing a variety. So that would be very challenging. And usually in breeding programs, we would be specifically targeting specific traits. For example, resistance to a particular pathogen or a particular pest or things like that. So in that case, you can be more targeted and you can limit your molecular markers to a few.
Thank you. I think that's a good question. A lot of back crossing has to be done. So you may have heard the news from ICRISAT India. They were able to develop the world's best or largest or the best heat-tolerant PGNP has been released about 2 weeks ago. So smaller traits and other things, breeders always deal with multiple traits. So then you bring it, and then you will lose some, then you back cross it, then finally you get it. So that's how they do, but these molecular techniques can be used. There are examples, but still long way to go. So thank you. I think without any further questions, please join me in thanking Sruthi to coming all the way from US and giving us the talk. So Sruthi is for her sabbatics 2 weeks with us, but she also done somewhere in Spain, so she's around next week, and she's also meeting PhD students tomorrow, some of the PhD students who expressed interest in meeting her. And certainly contact Cora if you want to meet her again next week, but with Ying Long, you will be meeting tomorrow. So once again, thank you very much. We really enjoyed. I have a lot of questions, but I won't ask any questions to you. Now, we have a number of things happening. So the next so-called special lecture is by Dr. Gustav Ginsberger. He has got a lot of experience on rangeland, and he has worked a huge amount of time in Syria, in Libya, and in terms of rangelands. And then he will be talking on the 16th of July, 2 p.m. to 3 p.m., his experience and wealth of experience, and see what implications for Australian rangelands. Then we have got our 20 years of industry forum. This is the 20th year, research, development, and extension investment in agriculture in Australia. Of course, they will talk about all RD&D investment in Australia. What will the horizon look like next 10, 20 years? I know it will change, and we are concerned that government is reducing investment, so the private, like industry corporations are coming, so that will be quite an interesting day, including our chief scientist. New chief scientist will be here. Fiona Wood, Professor Fiona Wood, Wood from medical professor is coming. She is in the committee in the federal government looking at RD&E investment in Australia for the next 20 years. And farmers and private breeding company and also consultants are going to talk about how should we invest RD&E in Australia and including Western Australia over the next 10, 20 years. So that's another important event. A Minister for Agriculture is likely to come. Then our Hector and Andrew Stewart Memorial Lecture. This is again a very important lecture, and that will be given by Professor Salah Sukaria from University of Sydney, and he's a great engineer, but he will be looking at integrating physical and digital agriculture, AI, drones, sensors, etc. I think it will be a very interesting day. That's on the 25th of September. That's 5:15 p.m. at the Bailey's lecture. And finally, I would like to, this is called the Professor Alan Robson AO lecture hall now. We will have his photograph coming. He was our former Vice Chancellor, the great Vice Chancellor. He passed away last November, and we, the committee, has agreed to establish two memorial funds. One is called Alan Robson Remote Scholarship, and he valued the students particularly from regional Western Australia, and we would be selecting a student from regional Western Australia to come and study in this university in any area, not necessarily in agriculture. It could be humanities, music. The second one is Alan Robson Lecture Series in Agriculture. So we wanted to have that a perpetual lecture ongoing, so you can scan the QR code, and any donation you will get, you will be able to do that. That's important. So we'll be looking at our alumni We have about 1,500 to 2,000 alumni who did agriculture science from here over the last 100 years. And that's it. Thank you very much, and have a great lunch. Thank you. Thank you.
Talking very well.